Hello, good afternoon, Ed. Thank you for joining in here. So let's start with the first session of the masterclass in genomics and nephrology. And in today's session, let us discuss the foundations of genetics and genomics. I'm Vinod Scaria. I'm presently serving as Chief Data Officer at Karkinos Healthcare and an adjunct professor at IIT Kanpur. Now, I'm sure you would be viewing this on the YouTube Live. Please feel free to put in your questions on the chat box on the side of the YouTube Live, and would be more than happy to take these questions at the end of the session. Now, before we start the foundations of genetics and genomics, I think I'll spend a couple of minutes to sort of go back a bit into history of something that has been happening very similar across the world. And in fact, if you really go back to the history, it's only around 50 odd years ago that humans could peek into the human body. And that has been pretty much with the discovery of what we today call as a CT scanner by Godfrey Hounsfield, who was later knighted for this invention. Now, in the last 50 years of a CT scanner, it has actually become quite a common investigation that is widely available and accessible across the world. Now, why did I bring a CT scanner in a genomic session? Is largely because of the fact that if you put this in perspective, it is only just around 23 odd years ago that we have been able to sequence a human genome. And in fact, the first draft human genome was announced in 2003, which first sequenced 3.3 billion alphabets. It costed approximately $2.7 billion. It took multiple organizations, thousands of scientists from 6 countries to be able to do this unique feat. Now, India was not really far behind in our ability to sequence the human genome. And the human genome is essentially a blueprint that we can peek into and which forms the basis of every one of our 37 trillion odd cells that form our human body. And it was in 2009 end that India sequenced its first genome. I was fortunate to be part of this team. And to this effect, India became the 7th country to be able to sequence its whole human genome. Now, to put this into perspective, we need to really look at how a human body is organized. And I'm sure you've learned this in your medical school. A human body is formed out of tissues, and tissues are in turn formed out of cells. And it's estimated that a human body, an adult human body, has around 37 trillion cells. And each of these cells has almost exact complement of the blueprint that we call as a genome, which encompasses 22 pairs of autosomes and the sex chromosomes. And all these cells were formed from the one single cell during embryogenesis by division to form, differentiate into different tissues with different functional correlates, which forms the human body. Now, when we start about this blueprint, I'm sure you have seen this in your textbooks, probably in your schools. And broadly, this is what our chromosomal karyotype looks like. The entire genetic material of 3.3 billion alphabets is organized into 22 pairs of autosomes, pairs because each of these of the pair is inherited from our parents, along with the sex chromosomes, which also forms or determines the biological or genetic sex of an individual, XX and XY, for a female and the male respectively. Now, what we need to keep in mind is that in terms of the genetic inheritance, every single base pair in our genome is inherited from our parents by virtue of the genetic material that was passed on by the sperm and the ovum, and by fertilization forms the first embryo, and the embryo divides, differentiates to form an adult human being. And half of this genetic material is passed on to the next generation by virtue of sperm or ovum, which are produced by the germinal epithelium. So, in many words, when we talk about inheritance, we need to keep in mind is that our genome is inherited from our parents. Our genome is also transmitted to our offsprings, or in a philosophical way, we are pretty much the carriers of this genetic information, which has been passed on by millions of years of evolution from generation to generation. It, we don't really own this genetic information because this information is shared with our parents, and we also pass this on to our offsprings. Now, having said that, the next big question is, what does this code contain? In very simple sense, if I were to take metaphors, this is pretty much like a recipe book. Now, assume that I need to make choco chip muffins today. I would go to the book that contains the recipes, and I would pick up the recipe for choco chip muffin and be able to follow the instructions to actually bake a choco chip muffin. Now, the genome essentially is like a recipe book, a quite large recipe book of around 20,000 recipes, each of them encoding for a particular gene, and the gene provides the instructions to make a particular protein. And the proteins are essentially the workhorses in the particular cell. And in a similar way, you could go to a library, take a recipe book, photocopy an individual recipe, and follow the instructions to create choco chip muffin. Your cell essentially does that, goes to the DNA, copies a piece of information as a messenger RNA, and the messenger RNA provides the instructions on how to make the protein along with the cellular machinery of ribosomes. Now, if that is the case, then we need to also go back into how this information is organized in the genome. The information in the genome is organized into discrete entities, which are called as genes. And there are around 20,000 odd protein coding genes and much more non-protein coding genes. And these genes have information interspersed with some characters which does not really make a sense in terms of information. And this information which is interspersed is called exons, and they are interspersed with information that doesn't really make any sense currently, what we call as the introns. Now, during this process, the exons, which forms the information, is spliced together to provide the instructions. Now, let's just assume that gene C has an instruction, and the instruction is Vinod makes red choco cake every day. It is interspersed with a lot of information, a lot of base pairs, which does not make much of information. The informational pieces, which are called as exons, are spliced together to produce a legitimate sentence, which reads, Vinod makes red choco cake every day. And if I can follow these instructions, I can pretty much go and make a cake. Now, what you need to imagine is that in the genome, which is quite large, around 3.3 billion alphabets, this information pieces essentially encompass only around 2% of the genome, less than 2% of the genome, what we call as exons, or what we call as protein coding exons, because this is the information that needs to be spliced together to provide the cells the instructions on how to make the protein. Now, the interspersed regions are what we call as introns, and introns doesn't much have information to encode for a protein, but it could have other additional functions, for example, regulating a gene or regulating splicing. And apart from that, there are also regions between these genes, what we call as intergenic regions. And roughly 98% of the genome does not really provide the information on how to make proteins, but nevertheless are quite important because it could have information that could regulate these genes and also allow this information to be spliced properly. Now, having said that, during the process of meiosis, which is an error-prone copying mechanism, your genome can have mistakes incorporated in the copying process. And this is very similar to what you do in a photocopying system, because in a photocopier, you essentially make a copy of the original. And the copy of the original is not always exact copy of the original, because you would have dots here and there, usually at random places. And this dot usually does not change the meaning of the text typically in the original copy that you photocopied. Now, this is very important to keep in mind, is because the information or the exons are only 2% of the genome. Most of these errors are not going to make much of a difference in the meaning. But once in a while, you could have single mismatches by chance, and this chance is extremely small. And these mistakes could either change the meaning or in some ways could also not change the meaning. For example, if you take the first case, instead of Vinod makes red choco cake every day, one single change could make it Vinod bakes red choco cake every day. Does not really much change the instructions, and I can still expect to have a red choco cake. But in a random process, this single mistake can sometimes change the meaning of the instructions. For example, if it could change to Vinod makes red choco coke every day, instead of a Coke, the cell today expects a bottle of Coke. And it doesn't really know what to do with this new stuff, because it was expecting a piece of cake and now it has got a piece of Coke or a bottle of Coke. And this is essentially what we call as functional genetic variations. Now let's move further and to also now define what a genetic variant is. And by definition, a genetic variant is a change in the nucleotide sequence at a particular position in the genome. And this is with reference or in comparison to a reference sequence or a reference genome assembly. Now, broadly, that means that if you have a reference genome, for example, if you have a build of a human reference genome, any change in any position with respect to the bases that are found in the reference genome is broadly what we call as a genetic variant. Now, genetic variations could mean many things depending upon the context, and it can come in very different forms. The commonest being a single nucleotide change. That means that at a given position, a nucleotide changes to another nucleotide. And there are only 4 nucleotides currently in the genome, A, T, G, and Cs. Now, in some cases, a single change can change the meaning or may not change the meaning, as in the case that we explained previously. Now, if it does not change the meaning, that largely means that that single nucleotide does not change the codon. And the codon are groups of 3 nucleotides, which provides the instructions to recruit very specific tRNAs and therefore very specific amino acids. So if the codon is not different, then you could produce the same amino acid, and therefore a nucleotide change could have no change in the meaning, and therefore we call it as synonymous or silent genetic variants. But once in a while, it could change the meaning because it could recruit a different tRNA because it's different codon, and this could change the amino acid, and such changes are called missense or non-synonymous genetic variations. Now, all these examples typically had a single nucleotide, but keeping in context the frame of the codons. But now assume that you could introduce a new nucleotide or delete a new nucleotide. This could call something called a frame shift, because then the context of information is lost. Instead of reading, since the ribosome reads in 3 nucleotide combinations, a change in a single nucleotide by insertion or deletion could change the context and produce a completely new set of amino acids. And this is what we call as a frame shift change, because this can produce a completely new sequence of amino acids which were not expected. Now, additionally, you could introduce a completely new 3-nucleotide insertion, and that would mean that the frame would still remain with an additional amino acid, and this is a non-frame shift genetic mutation with an additional amino acid by introduction, by insert, or by deletion. And once in a while, this change can introduce a stop codon, which will terminate the amino acid chain, and therefore these are called as stop mutations. Now, these are all single nucleotide mutations or single nucleotide variants, which means that only one single nucleotide at a given position is changed. It could either be silent or synonymous. It could change the amino acid, making it non-synonymous. It could change the entire frame of reading, what we call as a frame shift variant. It could introduce or delete 3 nucleotides in a stretch, therefore calling a non-frame shift variant. Or sometimes it could entirely terminate the chain and therefore called a stop genetic variant. Now, this is for single nucleotide variants. In some cases, you could have multiples of nucleotides or even large stretches of the genome, which could be either deleted, which could be inserted, which could be duplicated more copies, or it could be translocated from one chromosome to another chromosome. So broadly, there are very different kinds of genetic variations, single nucleotide variants being the commonest, but additional genetic variations which are also not very uncommon in the genome. Now, having said that, the obvious next question is how do we name these genetic variations? Because genetic variations are by definition context dependent and also dependent upon the reference genome by a genomic location, there are different genome or genomic versions available in history. And therefore, the obvious question is how do we port this information from one reference genome to another reference genome? And this used to be a big problem in literature. And therefore, there was a consensus built by a group of individuals who formed what we today call as a Human Genome Variation Society. And this entire consensus, which allow portability of information between amino acid positions or amino acid sequences, genomic positions, and gene level information, is what we today call as the HGVS nomenclature for genetic variations. Now, this allows to port information between DNA, RNA, and protein sequences. Broadly, what you need to keep in mind is that if you have a C prefix, that means the information is in the context of a cDNA or a messenger RNA. If you have a P prefix, that means that it is a protein sequence that is being talked about. And if you have a G prefix, that means that it is a genomic context that is being talked about. Now, the HGVS nomenclature provides an extensive rule-based system on how to really describe a particular genetic variant. And the commonest way is to essentially put it in the context of the genomic sequence. Now, for example, in this particular case, you have chromosomal 17, HG19, which is the build of the human genome, followed by G, that means the context is in the genome. You have a position which is 7578263, and at that position in HG19 build of the human genome on chromosome 17, the G position changed to an A. Now, this will allow us to put it in the context of the HG19 reference genome, chromosome 17, which is a portion of the genome, the genomic context of a very particular position, and look for a G nucleotide which changed to an A nucleotide. Now, this could also be in many ways put in the context of a specific gene, and that is what is described in the bottom, which is TP53 gene, which has a code which is called a RefSeq sequence code, which follows NM underscore with a number along with the version number, which says that particular version of the gene sequence for T53, the 586th position in the coding region, because it has a C prefix, the C changed to T. Now, there are tools which can actually port between a genomic location and a gene position. And, and I think this is something that you need to keep in mind, but you don't need to be worried about. Now, as we speak this, we need to always keep in mind is that a genomic variation is, while in the context of a position, this position does not really specify which of the two chromosomes does it belong to. Now, this is what you need to keep in mind, is also because of the fact that the both chromosomes have almost equivalent sequence. Now, if you have a genetic variant, which is only in one of the two chromosomes, then you call it a heterozygous genetic variant. And at the same position, if both the chromosomes have the same variant, then you call it as a homozygous genetic variant. And you could have two variants in a specific gene. If they are on the same allele, they're called as a heterozygous in cis. That means that if, for example, both variations were functional, it only affects one copy of the chromosome and the other copy is devoid of the genetic variant. Or it could be in trans, that means that both genetic variations are on the two complementary copies of the chromosome. Now, for sex chromosomes, we don't call it as heterozygous. We typically call it as a hemizygous genetic variation because, for example, in a male, you would not have an equivalent complement of the X because you have the Y chromosome in that position. So if only one X is affected, then you call it as a hemizygous genetic variant. But in a female, you could call it as a heterozygous if it is only one X which is affected, or when two X's the same positions, you have the variant, then you call it as a homozygous genetic variant. Now, assume that this is clear, then the next question is, what is a genotype? There's some jargons that we need to explain. A genotype is very simple. It is a sequence of the gene or genetic variations in this gene. So essentially, a genetic variation could be defined as a genotype. Now, of course, a genotype is important when you're looking for a phenotype. Phenotype is, in very simple ways, how the organism appears, or what is the phenocopy of that organism, or phenotype of that organism. Now, what you need to keep in mind is that these are non-deterministic. That means that not all genotypes will have a phenotype, nor all phenotypes would have a genotype. The same genotype can cause multiple phenotypes, and the same phenotype could be caused by multiple genotypes. But nevertheless, a genotype is roughly the genetic variations that you have in your genome, and the phenotype is the manifestation in the organism because or not because of that particular genotype. They're completely unentangled in many ways. Now, when we talk about phenotypes are very, very common. It could be anything like, for example, color of the eye, which is a very common clinical phenotype across. It is not a disease-causing phenotype because it is part of the normal variations of humans around the world. It could also be attached or free earlobes. As you see here, there are individuals with an attached earlobe, individuals with a free earlobe. I'm sure you'd all be checking whether your earlobe is attached or free. And in fact, this is also genetically determined, but does not cause a disease, but nevertheless present across the populations. The other one is, of course, the color of the skin, and in fact, quite variable across global populations. And this is also genetically determined by genes and genetic variations in the pathways which encode for melanin, its production, transport, metabolism, and presentation. And of course, the other commonest of the blood are the blood groups, which is the ABO blood groups, which are also genetically determined by very specific genetic variations in the ABO blood group locus across the genome. Now, these are also phenotypes, and we call it as genetic traits in some senses because of the wide variability and presence in generic populations. Across the populations, you would see this. This is pretty normal. This is also caused by genetic variations. Some of them may have clinical implications, like, for example, the ABO blood groups could have clinical implications. Even the skin color could have clinical implications, for example, in the predisposition to have UV-related melanomas. Now, having said this, there are some genetic variations which can be so severe in the outcomes, and this is what we call as genetic diseases. And I'm sure you have all seen what genetic diseases are. Some of them are quite very common in the population. Like, for example, what you see here, lipid deposits in and around the eye could be a signature of familial hypercholesterolemia. There are individuals who are tall or who are dwarf. This could also be because of genetic variations, like, for example, because of achondroplasia. You could have what you see here is the KF ring, which is essentially a signature for a copper metabolism abnormality typically seen in Wilson's disease. So there are genetic diseases. Some of these genetic diseases have caught imagination and has been in mainstream media as movies. Some of them are quite very common in the population. I'm sure you would have seen some of it, if not all of these conditions. Now, how do we put the genetic variations in the context of the genetic variation frequency and the phenotypes is the biggest question that we have to address in a clinical setting. Now, let us put genetic variations on a map, and this map has two axes. One is how frequent are these genetic variations in the population, and the second axis is the effect size. You can sort of imagine this as the odds ratio or the relative risk that given a particular genotype or given a particular genetic variant, what is the chance that I will manifest the phenotype? Now, in day-to-day general clinical practice, you would see very common disorders like diabetes, hypertension, obesity, so on and so forth. Now, is diabetes a genetic disease? The answer is yes, indeed, it is a genetic disease, but the genetic variations do not give predictability to the disease. In other words, you cannot today do a genetic test for type 2 diabetes. There are genetic variations, but have very small effect sizes. That means the predictability of the disease based on the genetic variations are quite very small. Therefore, you don't use a genetic test to diagnose type 2 diabetes. In fact, you cannot do a genetic test to diagnose type 2 diabetes because the odds or the effect sizes of the variations are extremely small. Now, on the other end of the spectrum, let us imagine of diseases which are quite also prevalent in the population, like, for example, sickle cell anemia. Now, if you find an individual with a homozygous, that means both chromosomes have the same genetic variant, homozygous HBS variant, then even without seeing the individual, you would be able to say that this individual would have sickle cell disease or sickle cell anemia. In other words, the genetic variant is almost always strongly predictive of the clinical phenotype, or the relative risk of the clinical phenotype with the genotype is enormously large. In other words, I can actually diagnose sickle cell disease just by doing the genetic sequencing or genotyping. Now, these diseases are not very common in the population. When I say not very common in the population, I will qualify that not as frequent as 30 or 50% in the population. They're relatively rare in the population. Therefore, we used to call them as rare genetic diseases. But the caveat being that in some populations, these rare genetic diseases could be quite very prevalent. Like, for example, thalassemia could be quite very prevalent in the Punjab population. Sickle cell anemia could be quite very prevalent in some tribal groups. And therefore, the global prevalence may not be used as a common denominator for these genetic diseases. So let's call them as genetic diseases, where the effect sizes for the genetic variations and the odds that the genetic variant will cause the disease is enormously large. On the other end of the spectrum, you have genetic diseases where the odds are enormously small. Or in other words, they are associated only to some extent, very small extent, with the phenotype. Now, the tools and techniques to broadly understand these are very different. For looking at small effect size variants with high frequency in the population, we use a technique called as genome-wide association studies. These are largely associations, very small associations. And since these associations are not typically usable in clinical settings because the odds are enormously small, we should not typically use them in routine clinical settings. These are very useful for research, to looking at the biology of the disease, but very useless in many ways for clinical decision making. On the other end of the spectrum, for diseases like hemophilia or sickle cell anemia, or for that matter, thalassemia, or many conditions very specific to nephrology, for example, like Alport syndrome or polycystic kidney disease, the odds of the phenotype being manifested is enormously large if you find the genotype. And therefore, you would use sequencing or a genotyping application to be able to diagnose the condition or do risk assessment for the condition. Now, give a context is that if you find an individual with an Alport syndrome homozygous variant, pathogenic variant, then you pretty much know that this individual in his lifetime has a very high risk of developing the features for Alport syndrome. I completely agree that not everybody might manifest the symptom. There are issues of penetrance, but almost everybody would have one or the other manifestations which are consistent with the disease, given that the odds is enormously high. Now, having said that, let us move on to what is broadly in the context of single gene versus multi-gene. Now, typically for single gene disorders, that means genetic variations in a single gene could explain the phenotype at very large odds. You have a very large effect size for the variant, like for example, sickle cell anemia or thalassemia, for that matter, Alport syndrome. These are what we call as monogenic genetic disorders. On the other end of the spectrum, you have genetic disorders where there are multiple variants, each of them with small individual effect sizes, but could have an additive property. Just because multiple genes are involved in a pathway, multiple genetic variations could provide very small risk. This risk could be added, and this is what we call as a polygenic disorder. Typical examples being diabetes or hypertension. And the idea of looking at polygenic genetic variations or polygenic risk scores is that you can risk stratify a population of individuals to be able to identify individuals at high risk. Now, then, therefore, this is useful at a population stratification mechanism rather than at an individual basis. An individual basis polygenic risk score may not mean much of information unless the individual's polygenic risk score falls in the top percentile of the risk profile. So if you plot the risk profile, you will have a bell curve like this. The extremes of this bell curve would be individuals with extremely high risk and individuals with extremely low risk. For those individuals in that bell curve, there is still some utility in risk stratification to be able to implement things at a population scale. But at an individual level, it may not really make much of a sense. Now, of course, you have disorders which are oligogenic. That means there are multiple genes involved. They are of intermediate effect sizes and of modest variant frequency. The typical examples would be pharmacogenomics, because there are multiple genes which are involved in the absorption, metabolism, and excretion of very specific drugs. And each of them could have additive properties. And therefore, these are what we call as oligogenic conditions. The present clinical practice largely focuses on monogenic genetic disorders because there's a very high individual effect size. Therefore, you can predict the phenotype based on the genotype, and also some way to oligogenic genetic disorders, especially in areas like pharmacogenomics. But increasingly, polygenic risk scores are being studied in a population setting, which is very useful for population scale risk assessment. but not very useful at an individual level for clinical management. In fact, most of the guidelines today refrain from using polygenic risk scores for routine clinical applications. Now, then the obvious then question is, why should we sequence or why should we do all this genetics? And the simple answer to this is, as we understand more of genetics, we also understand that many diseases that we routinely see across in clinical settings do have a very strong genetic underpinning. And to give an example of a set of papers that was published just last year, there are multiple publications which has looked at cohorts of individuals for any given genetic condition or any given common condition, like, for example, dementia or sudden cardiac death, or for that matter, even chronic kidney disease. There are studies which have shown systematically that a significant number of individuals in these cohorts do have a strong genetic causality. And this could be 1 in 5, for example, in dementia, 1 in 5, for example, in sudden cardiac death, and almost 1/3 of individuals with chronic kidney disease. Not just young chronic kidney disease, but across. These are individuals who have a genetic variant, monogenic genetic variant with a large effect size, and in many ways contributing either to the causality of the disease or in terms of modifying the outcomes of the phenotype or the presentation of the disease. And therefore, increasingly, guidelines have started incorporating genetic testing. And this is one such guideline. I'm sure you've all gone through this, the 2024 NKF guideline, which sort of suggests who are the patients, for example, in a nephrology setting, who should get a genetic testing. And if you really put these numbers across, you would realize that a significant large number of individuals in any cohort of individuals across specialties would now be eligible for genetic testing because of the fact that a significant number of them could benefit from this diagnosis and also be able to access therapies in some settings, modify the treatments in some settings, allow prognostication in some settings, or in some cases, even preventive modalities. Now, that begs the question of, in a regular clinical setting, when you cannot sequence everybody today, probably in the future you would do that, but today it is still not possible to offer a genetic testing for everybody. Can you triage individuals who might benefit from genetic testing? Well, broadly, the guidelines are there for some conditions, like, for example, there are NKF guidelines for kidney disorders. But broadly, what you need to keep, or what should really ring an alarm bell, is one, early onset disease, especially disease in the young. For example, if you find diabetes in the young or CKD in the young, with or without family history, because family history doesn't necessarily always be a predictor, but it is indeed a strong predictor in many cases. Multi-system involvement, atypical severity of the disease, or rapidity of progression of the disease, or resistance to standard therapy. All of these should ring an alarm bell and could, in many ways, provide a suggestion that this could be a genetic condition. And apart from that, there are also eligibility defined by genetic clinical guidelines, and also some scoring methodologies across specialties which have now been developed to sort of assess the prior probabilities of given phenotypes or given clinical presentations that can become genetic positive. So broadly, this is something that you need to keep in the mind. Now, when we talked about family history, one important thing that we need to keep in mind is that we need to record the family history with its granularity. And this is by far, one, the largest predictor of a genetic positivity. And second, most importantly, it allows you to identify additional individuals in the family who could benefit from a diagnosis, who could benefit from prevention, who could benefit from early treatment or prognostication. Now, broadly, I'm sure you know how to draw a pedigree, but broadly, you could draw a pedigree using this nomenclature or notations. A square one denotes a male, a circle denotes a female, A line between a square and a circle denotes mating. You mark generations one after another. In this particular case, you have a mating and two children, one male and a female. You could, in some cases, rarely could have dizygotic or non-identical twins, which is notated by a divergent line from one single point, or identical twin when you have a line between them. When sex is unspecified, you could have a rhomboid. You could mark carriers or heterozygous variants by marking half of it. You could mark a carrier for a recessive sex-linked condition with a dot inside. A death is marked by a cross line. Abortion or stillbirth is marked by a small circle or small circle or a square, depending upon the sex of the individual, if it is known. The index patient is marked with an arrowhead, and the consanguine marriage or mating is marked by double lines between the mating patterns. This is broadly that you need to remember, not necessarily that you need to absolutely follow this, but while drawing a pedigree, this is something that we need to keep in mind. Now, having said that, let us now go back to the chromosomes that we saw, and I will say that there is something missing from this. And this is missing from this largely because of the fact that over years, we are overemphasized on this picture, and this picture is largely from a textbook in schools. There are genetic materials which are not part of these chromosomes. And in fact, one of the most important component which is not in part of this picture is what we call as the mitochondrial genome. And the mitochondrial genome is in a small organelle that we call as the mitochondria. Absolutely important in the energy production in the cell. It has around 16,000 base pairs, extremely small to be visible on a karyotype, and encodes for around 37 genes and widely implicated in a number of disorders, including in nephrogenetic conditions. What you need to keep in mind is that mitochondria is almost exclusively inherited from the maternal side, and therefore it will always show a maternal pattern of inheritance. Now, having said that, today we have a wide variety of genetic tests. This wide variety of genetic tests today encompasses from one end of the spectrum, like karyotyping. The karyotype is what I showed earlier, which sort of is a visual representation of the chromosomes under the microscope. Then you have advanced high resolution techniques where you can stain very specific regions of the chromosome, like for example, fish. You could run copy number at a chromosomal level on arrays, what we call as an array CGH or a chromosomal microarray. You could look at copy number duplications or deletions using additional techniques like MLPA. You could sequence the mitochondrial genome, which is called the mitochondrial sequencing. You have repeat expansion disorders, not much implicated in nephrology, but more in neurology, which could be looked at a very specific technique called repeat expansion PCR. You could look at single nucleotide variations either by sequencing on capillary sequencer or on an NGS panel or an exome or a genome. In other words, depending upon the resolution, whether it's one base pair, hundreds of base pairs, thousands of base pairs, tens of thousands of millions of base pairs, you could have very different tools to look at these genetic variations. And this has been one of the biggest problems in genetics because the right selection of the test was extremely important because there were strong predilections of some types of genetic variations for some types of phenotypes. Like, for example, if you come across a patient with ataxia in a neurology clinic, one of the closest differentials could be repeat expansion disorder like spinocerebellar ataxia. Now, none of these other tests would yield a positive result because there is a very specific test for that particular condition, which is a repeat PCR. Now, therefore, selection of the right test in the past used to be a big issue. Now, what I will discuss in the later part of the series is that there is sort of a convergence happening, and that is exactly why we need to embrace genetics and genomics. Then the obvious next question is, what then is the difference between genetics and genomics? The earlier slide showed individual genetic tests, which essentially looks at single genes, single locus of genes, or single type of genetic variations in the context of inheritance. And therefore, this is what we call as genetics because it focuses on the study of single genes and how these genes and genetic variations are inherited. But of course, you could today sequence the entire genome. And in other words, you could go beyond that single gene to look at oligogenic and even polygenic traits and genetic variations, or in other words, put in the context much beyond the diagnosis. And therefore, we use genomics as an opportunity because it allows to study the entire genetic makeup of the organism, not just a single nucleotide or a single genetic variation or a single gene, which is in the context. Now, broadly, that would mean that if you were in a bar at night and you're walking back to your car, you dropped the key. You were drunk, so you would want to look where the key was dropped. You would typically gravitate to where the light is and assume that your parking lot had only one lamp. You would spend most of your time hovering around that one single lamp in search of your key. Now, that is typically what genetics is all about because that is largely information dependent. Because I know that a particular genetic variation causes a disease or a disease is caused by a particular genetic variant, I would rather look around that particular lamppost, which provides the right tools, right settings to look at that particular genetic disease. But now assume that you could light up the entire parking lot, then you would have a really unbiased view of where to look for, and you would not typically gravitate around one lamppost because you have a fairly good chance of looking across the entire spectrum of the parking lot to find your lost key. And this is essentially what genomics is all about. And genomics, therefore, is all genetics and more. And the reason why we need to do genomics is it moves the context much beyond the diagnosis because it can not just do the diagnosis very efficiently because it provides an entire view of the genome. It can allow disease classification and disease stratification. It can allow for disease management, treatment selection, like for example in pharmacogenomics. It can allow for disease prognostication based on additional genetic variations, what we call as modifier locus. And more importantly, it can allow for early diagnosis, intervention, and even prevention of a genetic condition. And increasingly, with a number of genetic conditions being amenable to early diagnosis, prevention, or even treatment, a genomic view, therefore, becomes even more important. It is just like asking a question that if I find a genetic variant, can I also plan the treatment based on pharmacogenomics? Can I avoid adverse drug reactions by doing pharmacogenomics? Can I look at the outcomes of the disease based on the prognostic factors for a particular genetic variant, and so on and so forth? And I think with that, I will stop this session, but with a few points that we need to keep in the mind. The first point that you need to keep in mind is invariance. That means that the genomic complement that you have from the first cell of fertilization to the entire human body is constant from birth to death. Or in other words, it is constant or invariant from womb to tomb. Of course, there are exceptions to this. I will come to these exceptions in the last two slides in this session. The second is covariance. That means everything that you have in your genetic material is also shared partly with your blood relatives. It's shared with your parents for sure. It is shared with your siblings. It is shared with extended relationship in both directions, both maternal as well as paternal lineages. And therefore, this information would mean that if you have a genetic diagnosis, that genetic diagnosis would also impact additional members in the family by identification of additional members who could be having the same disease at high risk of developing a disease. And more importantly, in endogamous or consanguineous communities, endogamy necessarily means that you marry within the same caste, tradition, religion groups, or in consanguineous communities where you marry within your own blood relatives. Therefore, the chances of your spouse having the same genetic variation becomes enormously large, and therefore the chances that your children will have a genetic condition also becomes enormously large. The fourth point that you need to keep in mind is that genetics will allow for identification of individuals who are at risk and also be able to predict future onset of the disease for monogenic genetic conditions. And at least in some cases, it can also predict the prognosis or the progression of the disease. And with artificial reproductive techniques and prenatal genetic testing, you can prevent the genetic disease in the next generation. So it is not just a diagnosis is important, but the diagnosis can also allow additional members to be identified, and more importantly, can prevent this transmission to the next generation with the appropriate tools, which is culturally and religiously acceptable. Now, having said that, there are a few exceptions that you need to keep in mind. The first exception is that every cell division or every mitosis has a rate of error that gets incorporated. Now, you don't need to be worried about this because the rate of error is extremely small, and also because the information component or the exons in the genome encompasses only 2% of the genome, most of them would not have clinical or functional implications. But once in a while, these are what we call as genetic variations introduced during every cell division or mitosis, which is happening at the rate of around 2.8 into 10 to the power minus 7 base pairs per mitosis. Once in a while could provide a functional advantage or disadvantage, which eventually could clonally proliferate. And there are some conditions, including cancer, which is because of clonal proliferation of such advantageous clones. So this is one thing that you need to keep in mind. The other thing is an accessory to this is the fact that since cells divide and there are genetic mutations which could accumulate during cell division, not every cell in the body could have, therefore, the exact same genetic variant. In some situations, you could have additional genetic variations during embryogenesis. And in embryogenesis, I'll urge you to go back to early embryology. There are three layers which are formed: the ectoderm, endoderm, and the mesoderm. And then there are organ systems which are formed from one or multiple of these layers. And therefore, you need to always imagine that you could have genetic variations which are restricted to one or more layers. These are what we call as somatic genetic variations or mosaic genetic variations, which could be confined to only few cells in the body. Now, in this particular case, you see what you see is segmental neurofibromatosis. Now, it has enormous implications in genetic testing because typically what the material you use for genetic testing is blood, and blood essentially comes from the mesoderm. And this could completely miss out such situations like segmental neurofibromatosis because the blood does not have the genetic variation. The genetic variation is limited to the few cells that form the ectoderm, and that too in a very small segment of the human body. So therefore, the right tissue for a genetic diagnosis would be biopsy of the affected individual, of the affected part or affected segment of the body. And this is something that you need to always keep in the mind, because in germline testing, we typically use blood as the starting material, and therefore this could miss out very specific somatic mosaic variants involving only specific layers, whether it's ectoderm or endoderm. Having said that, I will stop there and take questions. We are also the first generation of humans who are able to read our human genome in the 50,000 years of modern human evolution. And I'm sure there are many more questions. There's a small handbook that I've put together called Genomics in Internal Medicine. This is a basic level handbook which allows you to learn through the basic concepts of genetics and genomics. And of course, at the end of this course, we would also have a similar handbook looking at very specifically genomic case scenarios in nephrology. If you're interested, you could go ahead and sort of get this book. Now, having said that, let us also use this opportunity to address any questions if you have. You could post these questions in the chat box on the Google YouTube